So thanks for the introduction. So I'll be talking about mouse whole brain modeling. In this work, the question that we try to answer is, if we look at all the possible data sets that we can obtain on the mouse brain and try to integrate them in order to build a point neuron network of the whole mouse brain, how far can we go? How accurate of a network can we create? In order to try to understand this question, we developed a workflow. And the idea in the workflow is that we have different data sets coming in on the left. And then we first build cell positions for the brain. Then we differentiate the different cells into cell types. After that, we connect the neurons together. And finally, we can simulate this whole network. And the idea here was not to build a perfect model of the mouse brain, obviously, because there's no such thing as a perfect model. But rather to create a model which we can, at every step of the iteration, validate against other data, so literature data. And then this is an iterative cycle where we can always create a model, see how it performs in terms of validation, and then start over again to make the model better. So I have divided this talk into 3 parts. The first part is about creating cells and then differentiating them into different cell types. The second part is about connecting them, and the third part is simulating the actual network. So how do we create cells for an entire mouse brain? So the first data set that we looked at, so this is from the Alan brain. So these are these microscopical stains, slices, where it uses Nissl staining. So on the left, if this was higher resolution, you would see small bluish dots, which are the cells of the bodies of the different cells in the brain. And on the right, the Alan brain has annotated all regions in space, so that for each point in space, you actually know exactly in which region you are. And so this is not just one slice, there are several slices, and the Alan brain has aligned them. And we also redid this procedure, and in order to create a voxel data set. So the way it's done, you just need to take the grayscale values of the pixels of the images, and then quantify them into voxels. So this small animation shows how the voxels are created. Now this is not in 2D, obviously, although here we show it in 2D. This is actually a 3D data set. And the way we interpreted this data set is that it's basically, so the more cells are inside of a voxel, the higher the intensity of a voxel. So for example, if you look at the cerebellum, which is the part now on the right, the cerebellum has as many neurons as the rest of the brain together. So it's a very, very dense area. So you see it light up on this volumetric rendering. So what we did, we developed an algorithm, which is a pretty simple acceptance rejection algorithm. So what it does, it creates positions randomly in space, but not just randomly, but also by following the spatial density that we have here. So it has a higher probability of creating cells in a voxel that has a high intensity than anywhere else. And we run this algorithm, and we created positions and so on, until we create 110 million cell positions, which is the number that we get from literature for the entire mouse brain. So that you can see on the right. So now we just visualized every cell position as a small purple dot. And as you see, again, the cerebellum is on the right side, and it's like so dense that you can't even see through it. So if we just compare what we've created with the original Nissl stains, so on the left you can see the original Nissl stain, and on the right it's a virtual Nissl stain that we created from the generated cell positions. And we visualize each cell as a small bluish dot as in a Nissl stain. And you can see that the two look pretty similar. There are some differences contrast-wise and intensity-wise, but that's just because we have a specific visualization which is not the exact same as a real biological slice. But this comparison shows us that the overall regions are the same, and what we created is roughly similar to what we can see in a real mouse brain. Now these pictures are nice, and it's a good thing to visualize the neurons like this just to be sure that the algorithm didn't do anything wrong. But in the end, it's just a very qualitative comparison. So we wanted to be more rigorous than that. And so if we look at the whole brain, we can actually say for each region exactly how many cells we've created inside of it. And so what we did, we counted these cells, we calculated the cellular density per volume in each region, and then we started comparing that to additional literature values. So every time someone would do an experiment on a mouse brain, and then they would report a certain, this many cells per micrometer cube in a certain region, we would put that in a big data set, and we would compare it with our numbers. So we can see that, so some of our brain regions are very close to what people find in literature. Some of them are a bit further, but it has to be said that it's a bit tricky, because if we look at literature values, there might be two different groups who publish two different articles on the exact same region in the mouse, in the same genetic line. And sometimes the two values that they find, one of them is 10 times higher than the other. And so it's pretty difficult to actually do a comparison, because how do we know that we're wrong? And yeah, but in any case, we have a lot of regions to compare. So yeah, on the average, yeah, we can say that it's useful information in any case. And also, what's important here, instead of just looking at what we've created, we're actually able to quantify the error that we've made. So if we redo this iteration, we get some additional data sets, and we integrate them, and we recreate a point neuron, we recreate the positions of the cells differently. We might be able to say, hey, this time I got less error, or more error for that matter. So it's instead of just creating models, we can also try to say if how close they are to the reality, or at least to what people have found in literature. At this point, we have an entire brain full of cells, but the problem is that these are only cells, so we don't even know if they're glial cells or neurons. So we looked at these data sets, which are the genetic markers from the Allen brain. So I'm not going to go through the explanation too much, because I guess you guys had the tutorial yesterday on this, or before that. But essentially, each genetic marker stains a certain cell type which expresses a certain gene. Now, this can be a bit complicated. Sometimes it's pretty straightforward, so we used only one genetic marker to stain inhibitory cells, at least so far. We will add some more markers. And the green marker is an example of marker that stains a subtype of glial cells. And we've used actually 6 markers like that, different markers that we've combined together to have a global glial marker for the whole brain. And what we can do after that is we take all our cells in the whole brain, and then we take our data sets of glial, so the glial marker, and we again interpret it as a spatial density, so the probability of finding glial cells, because these are voxel data sets again. And we also have to additionally normalize this by the total number of glial cells in the whole brain, which we also have from literature, which is more or less 1/3 of all the cells in the mouse brain. And from that, what we can do, we go through all the cells, and basically for every cell, we say if it's either a glial cell or a neuron. And on the top right, the green dots, which have a pretty low contrast here, they're actually the glial cells, which we took out from the other cells. And on the bottom right, we only have neurons left. So as you can see, the fiber tracts are now cleared. And we additionally use this genetic marker for inhibitory cells. And by, again, normalizing it with the total number of inhibitory cells in the brain, we were able to also say for each neuron if it's inhibitory or excitatory. And the red dots in the image on the bottom right, they are the inhibitory neurons, and the blue dots are the excitatory neurons. So we now have a full brain full of excitatory and inhibitory neurons. We also have the glial cells, although for a point neuron simulation, we're not going to use those. But we could still use those if somebody comes up with a theory that glial cells contribute this much and this much in simulation. But so far, we just put them on the side. Yeah, so this is our full brain. Again, this is just a nice visualization of how we can just go through the brain and look at all the cells. So here the colors are, the green dots are the glial cells, The blue dots are the excitatory cells, and the red dots are the inhibitory cells, or inhibitory neurons. Yeah, maybe a bit less interesting than the fancy visualization is the table on the right with all the numbers. So here again, we can do a similar step as previously, but this time on the glial numbers that we found and the inhibitory and excitatory densities that we found in several regions. And we can also compare them to values found in literature. Now this is getting already a bit more complex because literature is a bit more sparse when it comes to more specific cell types. So it becomes really difficult to find for a certain region the density of say, inhibitory or excitatory cell types, and it's even more difficult if you go more and more into subtypes of cells. But again, the idea is to be able to quantify with numbers what we created so far. Finally, this is something that we always wanted to do, but to be honest, it's very complicated. So there are a bunch of other genetic markers in the Allen brain dataset. Here are just some examples. And some of them are pretty straightforward, but some of them just show certain subtypes of cells. And the problem is that many genes, they are expressed in multiple cell types, so you can't just say that this gene is equal to this cell type, because that gene could be expressed in several other cell types. And yeah, these categories are often non-exclusive. So we have all these markers, but to be honest, it's really complicated to use them to distinguish the neurons into several types. And furthermore, the method that we use, we always have this spatial density, and then we normalize it by the actual number of that cell type in the brain. And in this case, for most cell types, or maybe for granular neurons, we can do it. But for most other cell types, it's very difficult to say the exact number of cells that are in the entire brain of this cell type. So in the summary for this first part, so we use the initial stains to create cell positions. Then we develop this acceptance rejection algorithm to create the actual cell positions. Then we use the genetic markers to differentiate these cells into inhibitory neurons, excitatory neurons, and glial cells. And we can validate every step of this iteration. So in the end, we have an entire brain full of excitatory and inhibitory neurons. And so it's pretty convenient, and we come here to the second part. The problem is that we are interested to have a network which we can simulate, and this is not a network, these are just neurons. So obviously we want to connect them together. So this is what the second part is about. So the first data set that we looked at is the Allen brain tracer injections. So this is a virus that they inject somewhere in the brain, and it enters the neurons, and it follows the axonal projections of those neurons, and it will not cross to the next layer of neurons, at least in theory. And they have made thousands and thousands of these injections, so everyone on a different mouse. So these are microscopy slices, but they actually did the really great job of making available the voxel version of every experiment. So on the right, I'm just showing a few voxelized versions of these. So we only used, so they also have multiple mouse lines. We only use the wild type because some genetically modified mice can have absolutely different connectivity in the brain. So one has to be careful. Yeah, so here I'm just showing a few examples of, there is always an injection site where they inject the dye, and then there is always and then the dye follows the axons, and we're able to get that in 3 dimensions, and it's aligned to the previous atlas as well, which is really great. And the way we create connectivity out of this is that we take all the neurons in the injected site, so the first thing to appear, which is the figure on the left. So here I'm just showing one example with one experiment, but we do this for thousands of experiments. And we take all the neurons that are in the injected volume, and we start creating synapses that target neurons that are in the projected volume by following the probability of the tracer as a probability. And on the figure on the right, we see that we just visualized all synapses as small dots. And we see that, I mean, it's just to see if our algorithm worked. We're able to get a pretty similar distribution in space. And we do this until all neurons are connected. Now, if you look at the injection site here, it doesn't actually cover the entire, so if you look at all the experiments that they have done thousands, but still, all the injection sites combined, they do not cover the entire volume of the brain, unfortunately, but a big part of it. So there are some neurons that have not been injected at all, so don't know where the axons go. And for this, we apply the approximation of having an interpolation. So if there is a neuron that's between two injection sites, then we assume it belongs either to one or the other injection site. This is an approximation, and if we get more injections experiments, then we'll definitely have to do this much less. Yeah, so this allows us to connect at least, to connect our neurons together. And maybe the last thing that's important here is that usually long-range connections are mostly excitatory. And so for this, we only create outgoing synapses for the excitatory neurons. So the inhibitory neurons at this point are not connected. Yeah, we also did some validation on this. I'm not going to spend too much time on this. We compared our connectivity matrix, which is the second figure there, to the matrix that the Alan brain created from directly the experiments, so directly the injections. And there were two big differences in which we, instead of making a matrix from the injection experiments, we applied the raw data to create the network, and then we computed the matrix. And we also did this interpolation between the injection sites. And also the Alan brain removed a few elements of their matrix because they did some p-value analysis. So our matrix looks a little bit more blurred out because we have some connections that they do not have, which we inferred. Yeah, we also looked at several other distributions. At this point, it has to be said, it's pretty difficult to validate the connectivity that we created besides this one, because, I mean, even in this validation, we essentially just validate our neural network against the data that it came from, because yeah, it's very difficult to find entire brain connectivity for the mouse. Yeah, just one parenthesis, we also tried to do DTI, just because it's very tempting. After all, you have one data set for the whole brain instead of having thousands of tracer injections. So on the left, you can just see the fibers visualized in the whole brain. And on the right, this is just a 2D slide. So what we actually did, so in order to create connectivity from DTI, what we did, but we just tried this out at one point, is you take, for every neuron, you create a virtual fiber, which you let diffuse along the lines of the DTI. And for some part, this would work. So for example, if you have a neuron here, and you let some fibers diffuse from it, then it's going to cross the corpus callosum and get to the other hemisphere, which is usually what fibers do, at least between cortical regions. The reason we are not using this data set is that imagine that you want to connect this neuron to the opposite hemisphere. Now, if you look at AAV tracer, which is pretty precise, then what this neuron would do, as long as it's in the cortex, and what usually happens is that it would connect to the other hemisphere, but more or less at the same location in the other hemisphere. So it's pretty symmetrical. And the problem is, if you create some fibers and you let them diffuse through the corpus callosum, then all these fibers, they're going to mix together. And in the end, your fiber will have absolutely no idea if it should come out here or if it should come out here. And so for this reason, yeah, we did not use DTI, but this was just a parenthesis, because it certainly sounds very tempting to use it. So here we connected the whole brain. Now there are a few problems with this, though. The first problem is, if you look at the injection site here, it's actually pretty big. So it's a decent size. It's probably thousands, even millions of neurons that have been injected. And here we see the axons of thousands of neurons. And the problem is, if you're wondering about local connectivity, so if you wonder if this neuron and this neuron next to him are connected, you cannot find it out with this data set, because this data set would tell you that they are all connected if they're close to each other. So we only use this data set for long-range connectivity. And the problem is then the brain network doesn't have any short-range connectivity, so no neighbors connected to each other. And also, as I mentioned earlier, all these connections, they are excitatory. So because long-range connections tend to be excitatory, so therefore we have a big network, and it's only excitatory neurons. And yeah, if you ever run simulations like that, you know what happens. Basically, either you have an explosion of activity or no activity at all because you didn't stimulate it enough. But that's really not good. Yeah, also another thing which I haven't mentioned so far is that we connected all neurons here. So all neurons make long-range connections, which is absolutely not true because only certain cell types make long-range connections. So far we connected all of them, but this is something that we will change in the future, but we are totally aware of this. So we have to create local connectivity, and unfortunately there is no whole brain data set that tells you that local connectivity is like, you can't describe local connectivity for the whole brain because it's so specific to every brain region. Every brain region makes different morphologies of cells and connects with different directions. So the only way to do this is to go through regions individually, unfortunately. So the one we started with, since I'm in the blue brain, we have this very, very cool column. And it's very, very detailed, so all the morphologies are there. And also they have multiple cell types for each layer of the cortex. And so actually, since we create a point neuron model, we don't need this many details. So what we mostly did, we used the cell types. So we overrode the neuron type information that we had on the cortex, and we replaced it by these cell types. Oops. No, go back. Okay. And we also extract connectivity from this colony, and of course it's a simplified version because we don't have all these morphologies. And we created these matrices, which are essentially cell type to cell type connection probabilities that we can then apply to our neurons in the cortex. And also, we have a distance-dependent connectivity probability of connection. This is when the neurons are the closest, and this matrix is when the neurons are the furthest. So obviously it goes down. Now, there are two things that have to be considered here. First of all, it was a bit complicated because usually when people model the cortex, they assume that there is always the z-axis and that's always up, and then there is the x-y direction, which are the two lateral directions. And the problem is here we have a mouse brain shaped cortex. So we had to sort of compute the vectors for the entire cortex which show the outside direction. And the second part is that, basically this data comes from rat, and it's also barrel cortex. And we actually applied this for the mouse and for the entire isocortex, which is a bit approximative. But so if we get more data on specific regions, then we can just override this and use the more specific data instead. So this was for the isocortical regions. Just to shift a little bit, we also started work, this is still work in progress, started working on the cerebellum. So we have also additional cell types that we can apply to the cerebellum, which I'm showing a few here. And also we have some connectivity between the different cell types of the cerebellum. All these numbers come from different sources. So we're doing that in collaboration with the Neurolab in Milano. I think the take-home message on this slide is that really every region that we go through has a very, very different way of local connectivity also, but also the data that you find in literature is also very different. So for the cortex, we had this very, very detailed morphological column, and here we rather have these uniform connectivity numbers, which is, every region is described differently. So finally, so far we've done the isocortex and the cerebellum, but the problem is that the rest of the brain is not connected because we haven't gone through it so far, and that's still a very big excitatory network. So what we did, and this is very temporary, is for the remaining inhibitory neurons that are not in the isocortex or the cerebellum, we created this local inhibitory connectivity. So each inhibitory neuron would connect to its closest neighbors. So far, it's pretty simplified. It's a stepwise function. But this is very, very temporary, and we want to replace this as soon as possible. But this is just to say that we need to have something for the whole brain if we want to do any simulations, because it takes a long time to go through every single region and have some quantitative description of the connectivity there. So finally, on the second part, so we recreated long-range connectivity with these AAV tracers. Yeah, unfortunately, they don't have the enough resolution to create microscale connectivity. So that we have to get from individual regions. And yeah, we added this placeholder local connectivity, and that is to be replaced. And finally, the simulation part. So now we have a network, and obviously we want to simulate it, otherwise yeah, it's not that interesting. So this is the last part. So first of all, let's look at the numbers here. Now, first of all, this is a point neuron network. So we simulated it using the NEST simulator, which is really meant to run these large-scale simulations on even supercomputers. And so if you look at the numbers, you have 70 million neurons that remain, and you have 700 billion synapses for the whole brain simulation. And well, you're not going to simulate that on your laptop. So there are two limiting factors in a simulation, usually, at least the point neuron simulation. The first one is the number of neurons. And the number of neurons in a point neuron simulation usually limits the speed of the simulation. So because the simulator has to spend a lot of time just computing the membrane potentials of neurons. Whereas synapses, they're a limiting factor on the memory because they take up a huge space. So in our case, it's 10 terabytes of data just when we store the network. But the synapses do not contribute that much to speed-wise. I mean, they do contribute, but not as much as neurons, because synapses need to be only computed when your neuron actually spikes. So we run this simulation on the blue gene queue, which today I don't have any visualization for, but we also create these scaled-down versions where we reduce the number of neurons, so that we can run simulations on smaller PCs. Now you have to be careful when you scale down a network because you have to take into account how you scale down the synapses, the local connectivity synapses, and so on and so forth. So we usually don't trust these simulations that much for any studies, but it's nice because you can quickly see how the activity propagates. And usually, actually, if we don't reduce these numbers too much, the activity propagation on the scaled down version is pretty close to the one run on the blue gene. Yeah, I just have to give it some time. This is come on. Yeah, so this is a simulation of the brain. So this is a scaled down version. This is only 1% of the cells, the neurons actually. So all the little dots that you see illuminating, that's a spike essentially. And each color that you see is a different region in the brain. So for example, the yellow, that's getting very strong now, that's the cerebellum. And the pink, that's the thalamus in the middle of the brain, and so on and so forth. It's all just following the Allen brain annotation colors. But it's very useful to see single regions. And what you can see is that we're able to have some, so we actually do a stimulation on the primary visual cortex, as well as on the primary somatosensory cortex and on the barrel cortex. But the stimulation is mostly only needed at the beginning because then the activity just stays there. And just propagates and doesn't really disappear, which is great. Also, yeah, as I said, this is 1% of the neurons only, and this was actually run on this laptop. So it's pretty nice that you can make this scaled down version. And also what these visualizations allow us to do, well, first of all, we can just see how the activity propagates. But secondly, if we did any mistake in our algorithm at any point, we will notice it immediately that something is missing, or if we ship the GID somewhere, then we will definitely see it here. And it's a great way to debug code, and also, yeah, it does look nice. Yeah, just another smaller simulation. So on the left, we've run a simulation where we didn't use this inhibitory connectivity. So as you can see, it's pretty much an explosion of activity, so there's not much point to running it. Usually NEST, the simulator, also complains about this, understandably. But I removed the error messages. So. And on the right, that's with the local inhibition. Yeah, this simulation, I did it very quickly. This is actually even less neurons. This is like only 0.1% of the cells, which is really few. But as you can see, the activity is actually not that different from the previous simulation, at least the one on the right. And So what this allows us also to, what these visualizations allow us to do is to see how much connectivity affects the activity. Well, obviously, this one you would have noticed on the raster plot too, but yeah, it's pretty useful. And if we change the connectivity, we can always just compare the new activity propagation with the previous one. So let's just take a step back and look at what a computational brain simulation provides us. So first of all, we have the spiking activity, as I already showed. And here I also show it in a raster plot form. Again, the colors are region-wise. But in a computational simulation, you actually have access to a huge amount of data. So you can have access to the membrane potentials of all 70 million neurons in the brain at every time step. You can even have access to the synaptic currents at every time step, although you will never be able to store that on your hard disk because it would be 10 terabytes at every time step to store. But this is just to say that if we run a virtual neural network, we know everything about it, essentially. Whereas in experiments, when people do measurements on mice that are alive, it's very, very difficult to access its brain, and also you have to keep it alive and so on and so forth. So just the spiking activity is already impossible to measure for all the cells of the brain. You can measure it maybe for a dozen, maybe for hundreds. I don't know the exact numbers nowadays, but still, there is no way to measure it for the whole brain. So you can never compare it directly to the simulation. And usually in every experimental measurement, there is always a trade-off between resolution of the data that you get, spatial or time-wise, and the coverage of the brain. So if you record a single cell, obviously you're going to get everything, well, at least the membrane potential, but you can't record the whole brain. And if you have techniques like fMRI, which record the whole brain, then you're going to have a very bad resolution, both time-wise and spatially. So here I've just listed a few techniques that are often done. So the first thing that we found interesting was the voltage-sensitive dye imaging. So you've probably heard of this. Essentially, what they have to do, they put this voltage-sensitive dye on the top of the brain of the mouse, so on the cortical surface. And then the dye goes into the tissue. And it's mostly expressed in layer 2-3, although we have also modeled this. And then what they do, they put this camera on top of the head of the mouse, and they do some stimulation on the mouse. So here, what you see here, this is actually not a real VSD. This is actually, we computed this from the simulation. So every simulation that we run, we can get this virtual voltage-sensitive dye imaging from it. And we can compare this to voltage-sensitive dye that we got from literature. So we have a data set from Carl Peterson's lab at EPFL. And what he does a whisker stimulation, and then he records the response. And here we did the same. We just did a whisker stimulation, and then we recorded how this looked like. So I haven't put any comparison here because this is a slightly older voltage-sensitive dye imaging. But essentially, if we look at just the time constants, the time that it takes for the activity to reach certain region from one region to the next, and also the order of activation of different points on the top of the cortex, it's actually pretty similar to experimental values. Yeah, again, the one big difference here, we get much more activity than in the real experiment. That's why all the images are mostly red. But we can definitely compare these to experimental measurements, and that's great because then we can try to validate, at least try to quantify how much our simulation was close to reality. And yeah, secondly, we also thought about fMRI. So on the right, I just displayed this well-known hemodynamic response function. So this is usually what people use to get from neural activity to actual Bolt signal. And yeah, it has a pretty bad temporal resolution, and also fMRI has not a very good spatial resolution, especially in mice, because mouse brains are smaller. But also for every simulation, we can also compute the virtual fMRI of the simulation. So we get a 3D data set at every time step. So far, we haven't done any comparison because we haven't found any data sets of mouse fMRI, but we certainly want to do that. And also the way we compute the fMRI, the virtual fMRI, is that so a lot of people, what they do, they take the spiking activity of the neurons in a certain voxel, and then they just convolve it with that HRF function. In reality, it's much more complex. here what we do, so I'm not showing any animations of this because yeah, we don't have any data sets to compare it to. But we also take into account the synaptic currents that go into a neuron because that's apparently also contributing a lot to fMRI signal. There are also other techniques that I wrote down here, but so far we haven't tried to get a virtual version of those, especially that this is a point neuron model. So there are some of those techniques that you can't really compute, or it's very difficult. You have to make a lot of assumptions to compute it from a point neuron network simulation. So finally, yeah, we can simulate the whole brain model on a supercomputer, or we can do it locally, a scaled-down version. But again, we have to be very skeptical of the result of a scaled-down version. Although so far we've observed pretty similar overall activity. Of course, if we would look at smaller regions, it would be very different. Yeah, also we can visualize all of this to point out where we made some mistakes potentially, such as not having inhibition, and also. Yeah, there's so much data that we can get out of the simulations that actually there's no way to directly compare it to experimental measurements. So we have to process this data, and yeah, mostly we have to lose a lot of information. But yeah, that's what you have to do to compare it to experimental measurements. Yeah. So again, I would like to point out that we're not trying to create a perfect model. Because that just doesn't exist. Rather, we try to. So here already we've been iterating a bunch of times on the data set and trying out to create the model differently with different data sets. And this is very automatized, so we can just literally push a button and it will create a point neuron network and simulate it for us. And yeah, the idea is to refine this model until it gets better and better. And also the second important thing is that this is a huge mouse brain neural network that's lacking a lot of data in a lot of regions. So we're also really willing to so if anyone has a good description of local connectivity in this or that region, yeah, we are very willing to integrate it into our model and see what happens if it changes the activity, how it changes the activity. So we can see this as also some sort of a basic architecture that people can build, can add their models and build other models on top of. Finally, we have also made a collab on the HBP collaboratory. You have to be HBP though to access it, but it has this IPython, this Jupyter notebook, and all the code is already there, and you can just click a few times, and it generates you your own point neuron version of the mouse brain. Usually, it's a scaled-down version because this runs in a cluster, and it's pretty limited in terms of performance. But you can already go to well, numbers that are higher than usually what you can simulate on your own PC. And you can then download that and use it however you want. Finally, I'd like to thank these people. So there is Dan Keller, who helped out a lot with recognizing the different genetic markers and combining them into, for example, the glial marker that we created. There's Till Schumann, who did the implementation of the simulations and also another implementation of the network creation on the blue gene. And finally, there's Alberto, who's been doing a lot of work on the cerebellum part. So, thank you.